Hi, everyone, and welcome. I just want to introduce myself real quickly. My name is Catherine Mercica, and I am the Commercialization Education Program Coordinator at Fast Forward Medical Innovation, also known as FFMI. And I wanted to welcome you to today's webinar, which is number 5 in our 6-part series. So our last one will be next week. The Biomedical Innovation 101 series of webinars is provided by Fast Forward Medical Innovation, a unit of the University of Michigan Medical School's Office of Research in partnership with the Department of Internal Medicine. Before we get started, I have a few things that I want to cover, including some technical housekeeping items. If you have any questions that you'd like to ask today, please type them into the Q&A feature. Our staff will be using the chat function to answer questions and then share any additional links and resources. And the presentation is being recorded, and I will share out the recording and a PDF version of the slides to everyone, along with an evaluation at the end of the program. And with that, I'm going to kick it over to Brad.
Thanks, Catherine. Yeah, John Servos is on a very well-deserved vacation this week, and so Catherine is filling in nicely for him. So I appreciate everything John and Catherine do for the program. Again, for those of you that I don't know, it seems like I know most of the people on board here, but I'm Brad Martin. I'm the Director for Fast Forward Medical Innovation. And today's seminar is sort of a continuing series, and this one's going to focus on biomedical device development. And we're going to be discussing the processes for developing a biological device or development of a device, some of the commercialization and regulatory strategies that are involved. Michigan has a long record of developing medical devices. Actually, therapeutics and medical devices are pretty close in terms of numbers of patent disclosures every year by our faculty. So there's a long history of device development here at Michigan. And they run the gamut from everything from a very involved, highly technical prosthetic, robotic prosthetic, or all the way down to a 3D printed, very simple guide for helping implant subcutaneous implants and everything in between. But there's some commonality amongst these things when we're talking about medical device development. That's what we're going to be talking about today. And so with that, I will introduce our 2 speakers for today. First, you're going to hear from Dr. Eric Shah, who did his training here at Michigan, but has since left and gone to Dartmouth. But I'm happy to say he's going to be returning to the GI division here in the early spring sometime. Dr. Shah has been working with the GI division here at Michigan when he was here as a trainee to help develop a device to measure just synchronous defecation. But I think the lessons that he learns here and that he's going to be relaying to you are going to be applicable to a number of device development complications or technical issues that you may be facing. So we're very happy to have Eric coming back to Michigan as in general, and also happy to have him here today to speak to us about his experiences in developing that device. Then we also have our second speaker is going to be Adrienne Harris, who is the product development manager at a company here in town called Into Being. And anyone who is involved in the medical device space here in Southeast Michigan is usually pretty familiar with Into Being. I would venture to say they are the preeminent company in terms of helping individuals and businesses develop medical devices and bring them to market. They have a wealth of experience in developing devices, the quality systems that are required to develop a system. And so we're very lucky to have Adrienne here as well. And I think she's going to be really kind of playing off of Eric's points and his device development experience and talking about some of the common regulatory aspects or quality systems aspects to developing a medical device. Is that fair to say, Adrienne? Okay.
Yes.
And so with that, I'm going to turn it over to Eric, and who I'm, again, very happy to have him here today. And he can share his experiences in developing his RED device, rectal expulsion device. Eric, go ahead. Thank you.
Well, thank you, Brad, for the kind introduction. And it's really good to be, back in the fold with FFMI, and on this call today with Adrienne, and to share sort of what the experience has been, and with you all. So we'll start off by kind of going through this project, what it looks like. It's going to look very linear as this goes. I can assure you it has not been linear, and that is exactly what we'd love to discuss today. Just like science, when is it ever linear? So we're going to be discussing the rectal expulsion device to enable comprehensive constipation care for every gastroenterology practice. To give some background here, chronic constipation is more than hard and lumpy stools. There's also straining, sensation of incomplete evacuation, sensation that there's a blockage. So a lot of patients can have diarrhea, or so they think, and yet they're constipated at the end of the day. And constipation is an expensive problem. So there's a culture of undertesting with what gastroenterologists say might be the most appropriate treatments. There's a culture of overtesting. So multiple repeated colonoscopies, CAT scans, et cetera, generates about $8.1 billion in costs. There's 700,000 emergency department visits. If there are any ED docs on the call today, can attest to this, and 50,000 hospital admissions related to a quote unquote flare of chronic constipation. My goodness, at least 3.4 billion of this could potentially be preventable if only we did the right thing. So that right thing is defined over 10 years ago now by the guidelines from our American Gastroenterological Association, the AGA. So we start with an interview and a physical examination with the patient. We're going to consider a limited set of metabolic and tests, structural evaluation if need be, baseline labs. The patient is going to receive fiber and or laxatives for a therapeutic trial. And then according to the guidelines, when there's an inadequate response, meeting a gastroenterologist, we do a test called an anorectal manometry and a balloon expulsion test. This should theoretically be every patient that's seeing a specialty care provider. The balloon expulsion test is conceptually simple. So what happens is a balloon is inserted into the anal rectum, inflated with water. The patient then moves over to a commode or a toilet and is asked to poop. And if the patient can poop the balloon, that is a normal result. If the patient cannot poop the balloon or expel the balloon and it stays in, then that is an abnormal result. What we're looking for there are disorders of defecation. So there's dysenergic defecation. We can call it discoordinated defecation. But is it an anorectal issue is really the question we're trying to answer. This is the related test, an anorectal manometry. So this is a sensor probe that's inserted into the rectum. When the patient tries to poop, the anal sphincter should relax, which would be normal. Sometimes it doesn't, and it actually strains, squeezes, and the pressure goes up. And what follows then is that poop cannot come out. It's stuck in there. We call that dysenergia. Again, there are multiple names for that. If the test is normal and you do both of these tests together, then I'm not seeing an evacuation disorder of the rectum. I'm going to call it a transit issue, a motility issue. I can treat that. I have laxatives, drugs, different things in the wheelhouse. So this test has been useful. If the testing is abnormal, it suggests a defecatory disorder. And those are patients where instead of prescribing the patient laxatives and drugs and making the stool loose, I can send them to a physical therapist who can help improve coordination of the anal rectal muscles. And that treatment works. But here's the reality check. 2% of patients receive anorectal function testing. Among all individuals in the US with chronic constipation, this was a study of 1,700 patients across the US, accounted for sex, gender, age, geographic distribution, race, ethnicity, and only 2% of patients had undergone anorectal testing previously. 98% had not. What did their doctors say that weren't doing these tests? The guidelines are not being followed 98% of the time. They said, does testing actually change management? In my practice, where would I send my patient if I offered this testing today? Why should I test every patient coming to my practice for chronic constipation? This just doesn't make sense to me. So we'll follow the patient perspective. The patient starts off in primary care, sees a gastroenterologist. We do the best we can. There might not be local anorectal testing available. We're going to try the over-the-counter aisle. The patient's going to come back. We say, eat more vegetables, drink more water, prescribe laxatives, do a colonoscopy. We're looking for that kink in the colon, right? Prescription drugs, and we get a prior authorization. So we'll have a second colonoscopy. I really want to find that kink in the colon. Surgery, let's just get it out. Second opinions and repeated tests, phone calls, frustration. So a few patients are able to escape that. They go to a tertiary care center. They undergo anorectal function testing. They have a second visit to sit down and discuss the results, and that patient is then sent to a physical therapist. So it's a pretty inefficient symptom. And oh, by the way, there's a 6 to 12-month wait to get into a tertiary care center. So why not offer balloon expulsion testing today? It's a pretty simple test. So if we give this to gastroenterologists, this is what we actually thought we might do originally. We didn't know we needed to develop a device at the time. When we were going to give this to a gastroenterologist, they'd say, do I need to have a saline syringe, and how much saline do I put in there? Do I need to have a bedside commode in every clinic room? Now I need to have additional staff. And oh, by the way, I have 15-minute or 30-minute appointments. I already have 20 other things to do. Are my appointments going to be 5 minutes longer to account for that? So it just didn't quite make much sense. So we thought about this from a clinical workflow perspective and said, wouldn't it be great if we had a bedside test where we could streamline this process, make it easier, and help get the patient to a physical therapist straight away when there are 2 million patients coming with constipation to gastroenterologists each year. So we developed RED. So it simplifies balloon expulsion testing, enables point-of-care testing, provides immediately actionable binary test results. This is the device as it comes. And what happens is you unpackage it. It has lubricant and is inserted into the rectum. The end is popped off, the balloon inflates, and the patient is asked to poop. That's it. So we started prototyping this in 2017. We set out to do was build it cheap, build it with materials that are biocompatible, ideally, and then build some non-investigational prototypes, basically to turn it from something on a piece of paper into something that looks like it could work in the real world. We did this by a systematic review of diagnostic test accuracy to try to identify how the existing gold standard devices work and how to optimize their design the best way we could to inform how we designed our device. So what we found is that balloon expulsion test can be done in a seated position, but it can also be done in the left lateral. What is the left lateral position? It's basically the patient on their side right during and after a digital rectal exam. So we can do that right in the office, which was an interesting finding. And then we got the other parameters here that we used a little bit later on. So then we thought, we have an opportunity now, maybe not to build a water balloon to have patients expel, as is traditionally done, but instead, what if we could mimic stool and have patients poop out poop and, see what that shows? Fortuitous or lucky that we did at the time, we didn't know, but this was something else we did early on in the project. So after that first year, we spent 2 more years making devices, performing some initial biocompatibility assessments. I'm going to defer to Adrienne on discussing exactly what that is. It's very important. And, it goes to why these effective projects are most effective when there's a team around them. There are a lot of things that as a clinical investigator, I don't know. Reaching out and through FFMI really helped us along that path. So I'd love to spend a bulk of our time talking about that.
Eric, can I interrupt you and ask one question real quick?
Please.
So there was a device that was being used, but this was not being used very often, correct? You said 2% of the time. And so what were the key features of that device that existed previously that you did not like or that you wanted to improve upon? I'm trying to get to the point in which you decided we need to build something better. What did you not like about the current one, and how is yours improving upon that?
That's an interesting question. What it comes down to for us was a willingness to let go of the guidelines, and a willingness to say, hey, maybe we don't know everything and we need to pivot. And I'll be honest, that took a while. because, I think in the clinical sphere, there's a focus on the guidelines, and then the guidelines are not being followed. And therefore, we have to help educate people on following the guidelines. What we found was we did this customer discovery process where you have to go out and you go talk to a bunch of people.
Yep.
And what you learn is there's a reason that people don't follow the guidelines. And for us, that reason was that having a saline syringe, having water or saline or whatever you're going to use, it just adds these steps. And even if it's a few seconds long, grabbing a medical assistant or a nurse, somebody to help, it just eats up a little bit of time that makes this not fit into clinical workflow. And so it was, how do we take out all of those steps and design something that may not meet today's scientific perfection, but can really help with that clinical workflow in a day-to-day world. So, one of the things in the GI literature is interesting is that, when patients poop, the physiologic way to do it is when you're seated on a toilet, right? No one poops on their left lateral position, but the seated position doesn't make any sense in a clinic because the patient would have to go down the hall to a bathroom, which takes up time and it isn't pleasant if you don't need to do that. So, with a balloon expulsion device, so there are a lot of things that we learned in this process that really helped to inform not only the device, but how we tested it, how we've been testing it as we go. So yeah, it was customer discovery, I think.
Yeah. And I think the key point there is that for anybody who's on the call that's thinking about developing a medical device, you need to be cognizant of the fact that it has to fit in the clinical workflow. It has to be user-friendly. And not all clinical workflows are the same. The way we do it in Michigan is not the way they may do it in the Upper Peninsula. And so talking to people and ensuring that there is a broad customer base that wants this device to be put into the workflow is really important. Sorry to interrupt. Thank you, Eric.
No, absolutely. So I'll quickly go through our trial data just to show you, it's not so much the, scientific side of, what this achieved. It's, how did we think about this from the standpoint of, not necessarily commercialization, but really getting something like this out there? Like, what would be important for people to know at each stage? So the first trial was really designed around making sure that what we built matched up in some sense to what the gold standard represents. That was it. So we compared patients with chronic constipation, patients who didn't have chronic constipation, and just wanted to see if the results lined up between what we built and a traditional balloon expulsion device. The go, no-go decision, if any of you have ever submitted or thought about an SBIR grant, that's a key consideration. So that go, no-go decision would be, patients can't poop it out. Well, we got to go back to the drawing board. Something's not right. If everybody poops it out, it's not going to give us much information either. And we also don't want it to be painful. So we asked patients if they could tolerate it. So that's what our first trial was designed around. The last couple years have been, 3 years actually, been around the clinical use case. So if we gave this to practitioners, what they're going to want to know is, does the test change management? Okay, and that's it. So what we did is we did 2 phases. It was a feasibility phase where what we were really trying to do is figure out, what are the instructions for use of the device? Can we do a trial like this? How do we write down those instructions, how to use it, how to interpret it in a way that, anybody can do it. So all patients who enrolled in this trial had chronic constipation. They were a general gastroenterology population, which is important. It was not reflective of a tertiary care type patient population that's already seen 5 other people. This was intentionally to enroll patients that would be meeting a gastroenterologist on the first pass. So all patients underwent anal rectal function testing. All patients then went out to receive pelvic floor physical therapy in the community, which was important. So most of this was community-based pelvic floor physical therapy. Here's another schematic showing sort of how we designed this trial. So patients, we just made sure that they failed the typical fiber or Miralax for a couple weeks at least, and then they came back. All patients got tested. All patients went to see a pelvic floor physical therapist, and we did pre and post treatment outcomes. And what we came up with is you insert red into the rectum, remove the cap, and then the patient would sit in the left lateral position for a couple minutes. I might go chart and then just see if the balloon is still there 2 minutes later, and then come up with a game plan with the patient based on that. In the trial, we added a seated position to try to see if that also would help get data. The treatment protocol, and this was interesting. So we went out to talk to pelvic floor physical therapists, and part of the customer discovery is we just asked them, what do you do for a living? Tell us what you've done for the last 20 years. Is the treatment going to be reflective of what patients would actually receive for 2, say, million patients with constipation in anywhere? So the referrals were balanced across sites. The treatment was part of clinical care for patients. The treatment did include this called biofeedback. So it works on the anal rectal muscles as a primary treatment modality. The protocol was basically followed what they already do, which ended up being as the same as a simplified protocol that is a bit easier for patients to navigate. The traditional protocol calls for several visits to a physical therapist. The physical therapist here, much like anywhere, said, there's a limit to what patients will take. It's hard to do PT 5 days a week, without affecting other aspects of life and work. So we aim for about 3 visits with each physical therapist. The point was that the physical therapist designs our treatment protocol completely, and it basically followed what they already do. So we designed this like a trial. So we had a primary outcome to achieve a minimal clinically important difference in a outcome measure called PACSIM that measures global constipation improvement. So we could stratify if patients are responders or non-responders. We use the same sort of measure for health-related quality of life, responder, non-responder. And there is an FDA endpoint for drug trials around whether patients are able to have better stool frequency and form that doesn't measure the quality aspects of defecation that go into the primary endpoint. But we adapted the drug trial outcome just to get that information at this stage nonetheless. So the analysis was abnormal versus normal test result, responder versus non-responder, and we could calculate an area under the curve. Okay. We use this, it's called a Youden index. We could use this statistical method to figure out how you would optimize how to interpret a test like this to inform what you would do in practice. So of the 60 patients, we had 52 that were included in our intention to treat analysis, meaning that they attended at least one physical therapy appointment. So we had 88%, I think the numbers work out to 88% adherence. And what's interesting is that at baseline, 1/5 of the patients were pooping at least 3 complete spontaneous bowel movements per week. They were pooping, but they had sensation they couldn't get the stool out. They were seeing a gastroenterologist for constipation. So it just underlines that constipation doesn't just mean hard, lumpy stools. There's one adverse event. So anal pain with a suspected anal fissure. That pain resolved immediately, was replicated when the patient underwent anal rectal manometry, and then also with pelvic floor physical therapy, so anal fissure. And that is expected in this clinical disease entity. no serious adverse events. So baseline demographics here were similar to what we see in chronic constipation trials. It was reflective of what a general gastroenterologist should expect to see. We did get baseline interrectal manometry testing as well, along with that. And what we found is that the test was able to predict clinical outcome. Our areas under the curve are between the 0.6 to 0.7 range, which was intentional based on our power analysis. We were not aiming to identify a test that could 100% predict that a certain group would have a 100% rate of success with pelvic floor physical therapy compared to a 0. So because we thought that the responder rates and placebo and all of that might be smaller, the resulting AUC out of this was expected to be between 0.6 to 0.7. So it did meet on global symptom response, health-related quality of life, and then also this bowel symptom response in the left lateral position. It wasn't powered to do that, so it trended towards demonstrating that it might work in seated position, but we can't claim that because it wasn't powered to do that. So in the seated position, all right, how would we define a positive test? Who am I going to send a PT? So if we retain the device for at least 13 seconds, okay, in a seated position, I would expect a quarter of the patients I see to test positive. Then I would refer that patient to a physical therapist, if this device were approved, and we'll talk about that. Sensitivity and specificity are here, higher specificity. Interestingly, so an abnormal result would mean that I could tell a patient, I think you're going to have a 71% chance of responding to pelvic floor physical therapy versus a normal result says 28.9%. So there's a p-value there. It's statistically significant. In customer discovery, we learned that gastroenterologists like the 28.9%. They're going to refer that patient to pelvic floor physical therapy too. Okay, so yes, you can predict who's going to improve with pelvic floor physical therapy, but I'm still going to send most patients. The reason is that prescription drugs and other treatments, laxatives, have a 30% rate of efficacy anyway. So the paradigm works, but it wouldn't change practice. So the left lateral position was the most interesting. So here, if the device falls out within 5 seconds, or they retain it for more than 2 minutes, then 3/4 of the patients will test positive. So most of those patients, I would send to pelvic floor physical therapy just to try it. So we have a high sensitivity test. What's interesting is that with a normal result, so if the device takes a little bit more than 5 seconds to fall out, but isn't retained for at least 2 minutes, those patients don't respond to pelvic floor physical therapy. So I can help that patient not go to get internal rectal physical therapy and try a different treatment who would otherwise be willing to go. So that fits with what the use case would be in a general GI practice, and how they would like a test to perform. And so that was very interesting. So, just kind of spark the discussion here. One thing we learned, does the idea we have solve a problem that people actually care about? Okay. We think yes, right? The answer is probably no. So, what we developed originally looks nothing like this. And, I don't think we knew what we were going to develop. And that was entirely the point, that it took going out and talking to people who aren't quote unquote experts. But, there actually are experts. And so that reflects most of clinical medicine, so learning that, what the day-to-day life and challenges are like, even though we think we know it, it was really helpful. And even then, is it sufficient for investors? Is there a business around it? Would the FDA care? Or would it not be good enough? So there's this 3-question approach to commercialization. So I think John Servas and Jonathan Fay, led a course several years ago. And so, this was from what Jonathan Fay had presented. Is there a value prop? Is there a viable commercialization plan? Is there a return on investment? Okay. That 3-question approach, drilled over and over and over, and rinsing and repeat until you find something that might work. I think this would have been a fun science project if not for, meeting Adrienne and working with Into Being, being willing to pivot. That's where, we might have a great idea. The engineers know if that's actually something that's going to work, I think. So the rapid care pathway for constipation, as we would potentially frame it now, would be that a patient would come to a gastroenterologist for constipation or to an open access unit for a diagnostic colonoscopy for constipation, which, by the way, is not reimbursed for that indication. Chronic constipation does not cause colon cancer. Change in bowel habits might, but chronic constipation does not. The first time you get a colonoscopy, fine. The second, third, fourth time, I have questions. Okay. So the patient could be seen in the office, undergo red, a normal test, optimize the bowel regimen, abnormal test. We engage this evacuation disorder paradigm. So, what I think was really cool. So, my mentor on this is Dr. Bill Che. He's the chief of GI now. The way things are going in medicine, how do we help patients get to the right treatment faster? How do we create these clinical care pathways to do that, within these big black boxes, that we have in medicine? And then how do we leverage that clinical infrastructure that is wonderful with new medical technology to get there? And so that's sort of, the things that that fall in line with this project and with others that, I'm really looking forward to in coming back, and it's going to be a really exciting time. So this is, probably up 2% of the ecosystem in the university and in Ann Arbor. There are a lot of great people and resources, across the university and around Ann Arbor. So Into Being, there's a robust venture capital presence. There's Spark, there's incubation for incubator, and then FFMI and innovation partnerships I felt like has been a hub to learn and sort of navigate all of this, because there's a lot out there, and which is really cool. So here's our contact information. So you have my email. We have Adrienne's email here. Yeah, we have a lot of time left. This is great.
Well, thanks, Eric. I very much appreciate it. And I appreciate your kind comments too about the course and the value of that you found in that course. And just for everyone's information, that course is still being run by our office. It's called the Fast-Paced Program, and it's very much focused on customer discovery, customer segmentation, figuring out your value proposition, and coming up with a go, no-go decision on your device idea. So we do run that course still, and it's a very impactful course for a lot of different people. Catherine just put the link in the chat feature there. So that's another version will be coming up this spring, another cohort. We've run that almost 18 or 19 times so far. So Adrienne, I didn't know if you had any comments on just sort of not necessarily related to this project necessarily, but you've worked with a lot of U of M innovators on a lot of medical devices. Just general comments on device development, regulatory considerations, et cetera.
Sure. Make sure I unmuted myself this time. I'm going to use this project as an example because, Dr. Shah has laid out this beautiful development and science and how to get a device to show value. It's had a lot of really unique regulatory challenges for something that seems so simple, very low risk. All that put together, it's had a unique path through the FDA so far. There's also been unique opportunities for this device in that, as an example, it's considered a non-significant risk device, so it allowed the clinicians to get their device to a clinical trial on a speedier pathway than if it fell into significant risk. It's a non-sterile device, so you don't have to prove sterility before it touches patients, all that great stuff. So we were able to get science moved forward while mucking through the regulatory side with the US FDA. So a little bit on that. The first time we interacted with the FDA was with a 513G, and that was to get buy-in or feedback on what the actual product code and regulation number this device should fall under. And looking at what's out there, a lot of the devices that do balloon expulsion testing were kind of shuffled in under the bigger anal rectal manometry systems. We looked at this and went, this doesn't have a big electromechanical system to go with it. It's just a balloon. There's a product code and a regulation for balloons that get put in people for diagnostics. We want to pitch to the FDA that this should really fall under that. Dr. Shah reminded me that this 513G did go in during the government shutdown a few years back. I don't know how much that had a role. And then don't worry, after that, we rolled into COVID, which also drastically changed interactions with the agency. They came back and said, no, we don't believe that you're just a diagnostic balloon. And I'm going to read this just a little bit now that you've seen the device and you saw what it does and how it works. They said, here is the regulation you fall under. A motility monitoring system that is used to measure activity and pressure in the stomach or esophagus by means with a probe and transducers. It's introduced through the mouth. It goes in the gastrointestinal tract. It includes signal conditioning, amplifying, and recording equipment. It could have certain accessories such as pressure transducers, amplifiers, and external recorders. You're a class 2 device, go through the 510K process. Did you see any of that on the device? No. But because the predicate devices that do balloon expulsion testing came with the systems that do all this other stuff, and according to the 510K process and the FDA foundations, that's the category they wanted to put us in. Okay, we can work with that. The follow on to that was a pre-sub to say, all right, you put us in this classification. We've worked hard to find a device that could be a predicate. However, we want to check with you that all these things that we're going to come into your predicate system with, the point of a 510K is to show substantial equivalence to something that's already out there. All these other systems have big electromechanical equipment. They do have pressure transducers. They do signal processing. They do sensing. They do recording. We don't do ours. The unique challenge in that is the majority of the devices that are used on the market right now were cleared through the agency 30 plus years ago, which means you don't have access to the 510Ks to look at even what they did clear. It was a challenge. And the FDA would like you to use a device. It's a predicate that is within 10 years old at this point. They don't like you bringing something on to predicate to something 30 years old ago. We found a device that was recently cleared in the late teens, 20 teens, that had a balloon portion that did not hook up to all the transducers, did not hook up to the electronic system. And we went to the FDA and said, would this work for a predicate if we just predicate off of this portion of the system? And gave them all the justification and whatnot in that. And they said, yes. It's like, fantastic. We have something to go on. What was interesting though, is in that particular device, they don't actually have an indication for balloon expulsion testing. It does everything else. It looks exactly the same. It falls under the regulatory requirements and product codes and regulations, but doesn't have the same indication. So that's another challenge in how we were going to present the device to the FDA when it does go through the 510K process. And then that's where we'll get to tie in all this clinical data that the clinicians have been able to gather in support of it, which is what the FDA asked for in the pre-sub. Okay, you can show us all this stuff with bench testing, that it's very similar to the predicate, but we need to see some clinical data based on the differences that it does do the same thing. And this is all in the background. Well, actually trying to develop the device. It's figuring out how you're eventually going to get that device to commercialization through the regulatory challenges of the United States FDA. And that's kind of where my work has come in to help more so even on the development side, though we did start with the development of the device at the beginning. To my anecdote there is, this is the first time I've ever had to do force testing on what is considered healthy stool. That was a unique process in and of itself to determine what actually healthy stool behaves like. So it's been a fun ride all the way through. You are on mute. I do that all the time.
Maybe just for the benefit of those who are perhaps very naive to the device approval process, could you just sort of tell them the difference between a de novo pathway and the 510K pathway? You talk about predicate devices, just so people understand what you're talking about.
Absolutely. One step above that, the FDA has 3 classifications for devices: class 1, class 2, and class 3.
Yep.
Class 1 is the lowest risk. It means you do not need the FDA to review anything about it before it goes to market. Make sure you follow the QSR, which is 21 CFR 820. You meet all the requirements within that regulation, and you may go to market with your device. If you cause problems, the FDA is still going to come look at you. They can still inspect you at any point, but they don't have to authorize your commercialization. I'm using authorized because now we're going to get into the details. The 510K is for class 2 devices. The 510K is based on there being another device out there that is very similar to yours that you can use as a predicate. And what you're doing with the 510K is you are demonstrating substantial equivalence, not the same, but you're substantially equivalent to something that's already on the market. And if that's already been proven safe to be out on the market, then if you can show enough similarity to your device, then you can be safe. 510K, another alternative name for that is a pre-market notification. And if the FDA grants it, you get clearance. So not approval per se, and you don't get clearance or approval for a class 1. So you get your 510K cleared, and then you are cleared to go to market. Class 3, highest risk devices this is called a PMA, or a pre-market approval. This is when you would actually use the approval word device. I'm going to skip over that one because it's outside of the scope. De novo is very interesting in that it is for what the FDA considers automatic class 3 designation, but where you go in and then they review it to the point where maybe you aren't actually a class 3. And where this comes in very useful and kind of cornerstones with the 510K process. Let's say you have a device that from a risk-based perspective isn't a class 3, it's not a class 1, it really should be a class 2, but there's no predicate out there. If you don't have a predicate that you can take to the FDA to go through the 510K process, then you become a de novo. And a de novo, when you're done, you get a classification and a regulation number. Maybe you already fit into a regulation, but you don't have a classification yet for a device. Once that device is granted the de novo, if it is granted a de novo for a class 2, other products, including products that you develop afterwards that are very similar, can use your device in the 510K process, but it needs to have that initial foundation device to say it's allowed on the market as a class 2. The unique difference between a de novo and a 510K for a device that's ultimately going to be identified as a class 2 is de novos require clinical data. This can be a major hurdle for a system or a device that doesn't seem like it should have that higher risk, doesn't seem like it should need clinical data because if it went through the 510K process, you would not need clinical data, but that is the big gap between going through a 510K process through de novo. Here, if the FDA came back after our pre-sub and said, no, you know what, all these other products are too old and you don't match them enough, you need to go to de novo, not a huge punch in the gut because the team was already getting clinical data. But if you weren't planning on that initially, that's a huge cost that needs to be taken into consideration for your development process.
Great, thank you. Any other questions from the group here in our final 10 minutes for either Adrienne or Eric? Just go ahead and take yourself off mute and don't be afraid to chime in and ask a question. Medina, I know you had a question down here in the Q&A. I don't know if it's been adequately answered yet. It must have been. Going once, going twice.
So
Oh, a couple more questions in the
Oh.
chat. From Charles Schuler, probably to Eric. How did you find the commercialization process?
Yeah, it's interesting. Hard but fun. And it's a different sort of set of questions than the usual sort of scientific inquiry, but it's very similar. It involves really answering that question: do people care enough to care with money? And, you know, and that goes to impact, you know, does your, you know, paper, your project that you're publishing, you know, is it generalizable? Is it novel? Is it rigorous, citable, you know, all that. So it actually is quite similar in that sense. You know, the key is who's the audience. The audience is not going to be key opinion leaders in your field per se, because, you know, if you're, you know, on this call and thinking about commercializing an idea, you really want to develop that idea around so-called non-experts. But if you flip that, the non-experts are experts and their challenges are very different. And so that's really where I think the commercialization process comes in, is learning how to think about, you know, what that means day to day, which was, you know, which was really cool. And it's a process. That's what I think the fast-paced course was, you know, particularly helpful for.
And Eric, while we've got you here, one of the participants, Medina Sokolov, is asking you about your career stage when you started doing this work. And did that make a difference in terms of what you were able to achieve or not achieve?
Yeah, that's a good question. So I started this when I was a clinical fellow in GI in 2015. And so, like, before this work was a year of learning about the commercialization process through FFMI. And there were a series of grants. There was a GI Innovation Fund at the time, and they had 2 tiers of grants. And, you know, I think that in learning through the FFMI courses, really gave us the skills to learn how to be successful in the grant side for that. You know, and then since then we've, you know, had some professional society funding for this as well. You know, I will say there is federal funding opportunity for this type of work, either in the SBIR programs. And then, you know, you could use traditional pathways for it. You have it, you know, it becomes tricky if you're at the stage where, you know, this is a company per se. But, you know, that's where there are mechanisms out there, you know, at different stages to do this type of work. And, you know, I think FFMI is good, a great resource for that as well, because they'll know everybody who has, you know, been not successful in certain ones, which can help, and successful in other ways as well. So.
And that kind of gets into another question we had here from someone else asking about the funding available through either Fast Forward or Into Being. And a number of years ago, we used to administer a program for the GI division, which was made possible by a grateful patient and a gift that she made. And that's how Eric came about working in this space. But there are a variety of funding resources that exist on campus that we can point you to. Some are administered by our office, some are through the Coulter Foundation or through the Tech Transfer Office. Well, we can certainly put you in the right direction. Eric's comment about SBIRs, I often hesitate to talk to people about SBIRs because I think that forming a company and going through the process of forming a company for the sake of getting funding is sort of a bad reason to form a company. Let's form a company when it makes sense to form a company. Going after SBIR dollars is not the best reason to form a company. There are other resources on campus that allow you to de-risk a technology or a device or build a prototype or do some initial clinical testing before you get to that stage of forming a company. And that's exactly what those translational funds that are available to you through the university are made for. They're non-dilutive. They're just research dollars that are for you to develop a clinically viable product. And one of the other things I want to mention too is that we can do an awfully good job of doing some matchmaking between clinicians who have identified a problem and engineers who can build it. Don't think as if you have to be a jack of all trades as well as a clinician to build a medical device. We can do matchmaking between colleagues that are at the College of Engineering who have a lot of know-how and technology, but they're just clamoring for a clinical indication to apply it towards. And there's also people in the local area like Into Being and Adrienne who can help as well. So if you have any questions about funding, just know that there's a lot available, and come to our office and we can point you in the right direction. And many departments have internal funds. There's the Franklin Innovation Fund, there's MTRAC. There's a new program that's being put out by Steve Kunkel in the Office of Research called Bold Science, which we're trying to fund some outside the box ideas. So a lot of it is available.
To answer on Interbene's part, we don't provide funding opportunities specifically, but we have collaborated with a lot of researchers at U of M to help develop some of that writing for MTRAC, Kickstart, other types of grants, whatever you're going for, to help lay out a development plan that is made by people that develop medical devices. Again, we don't expect a GI doctor or a cardiovascular surgeon to understand the whole medical device development process. You have a great idea. And what we do is we can come in and help lay out how to get that idea to the next step, how to get it to the last step, and then also work with the team on that throughout the course. So that is one thing we have worked in it. We've worked with on the red device in that regard too, helping get funding for it as it's moved through the process.
And the one thing that I can say is with absolute certainty is that in my 10 years of reviewing these translational research applications for funding, you can very much tell somebody who has spoken with Interbean or has been through an educational program and understands the process. As the language changes, their understanding of the market and the path to commercialization becomes much more solid. And so taking a course is usually your first step. Talking to somebody like Adrienne at Interbean is usually your first step. And that really enhances your chances to get funding downstream. So, and there's the link for Interbean. Thank you, Catherine. Any other questions here? We're getting to the end of the hour here, and I want to be sure to respect everyone's time. Are there any other questions? Catherine, can people join in by audio, or is it just through text?
I think it's through text, but or chat. Well, let me think.
Maybe that is not the case.
I think I can individually go and let people talk. So, yeah, you should be able to, I think, raise your hand if you want to.
One last thoughts, comments, or questions? Otherwise, we will let you go. All right.
Okay.
Adrienne and Eric, you have our eternal gratitude. Thank you so much for taking time out of your busy schedules to present today. Again, if our faculty have any questions, they can either contact us or the speakers directly at the links that were provided. And hope you all have a great day. And thank you again to our speakers. Okay.
Thank you.
Bye-bye.